Thank you, Valeria. Thank you to the organizers for inviting me. I'm Brazilian, but I'm 95% Italian, so I always feel, genetically, so I always feel at home here. So I'm going to be talking about gene therapies in primary mitochondrial diseases. As you already know, mitochondrial diseases are complex because they affect multiple tissues. They have two genomes causing mutations that cause problems. So it's rather complex. And there are different approaches, as Carlo mentioned before, to treat mitochondrial diseases. You can have a more general approach with drugs that affect different, energy production as a whole. And they tend to be more general, usually only partially or not very effective. Sometimes they do help, though. But gene therapies have the potential to be more effective because they address the root of the problem. So if we can actually succeed to make this work well, it will be tremendous. So I'm going to talk a little bit about a nuclear DNA gene replacement. So mutations in nuclear DNA usually are approached by replacement of the defective gene. And then the mitochondrial DNA, the approaches are either you eliminate a mutant mitochondrial DNA in a heteroplasmic environment, or now you base edit a mutant mitochondrial DNA. So the nuclear DNA, I'll just give an example to discuss one specific point that I think it's potentially important. So we have used this mouse model that has a defect in NDUFS3, only in the brain, the cortex, CAM kinase 2 alpha neurons, and the mouse, does develop a brain disease. So what we did here was to try to preclude the development of this disease by putting back the missing gene when the mice were relatively young, using this AAV called PHPEB that is pretty good in getting neurons of mice, C57 mice, to express the gene that you put in the virus. So in this case was the missing gene, and the control was a GFP. So this was injected, and then we followed the behavior, and then we killed the mice at 5 months and analyzed the tissues. So you see here that the blue is when we inject the virus with the NDUFS3 replacement, and the mice pretty much, they live a normal life. This is a rotarod showing that the mice fall when they get the GFP. Those are the green bars. So it's the time that they can stay in the rotating rod. But when they get the blue, that's the replacing gene, they do pretty well. So the mice that got the GFP virus, they have this defect in complex 1, you can see here, but when you replace it, you can see that it gets stronger. And this is the quantification. If you look by immunostaining, you see here the knockout, there are lots of neurons without NDUFS3, and when you put back, you see that some neurons come back. But that's the point I want to make here. Not every neuron gets to express the NDUFS3, but still the mice get better. This is just to show that, again, these knockout mice, they get gliosis with increased GFAP and microglia EBA1, and that is restored to a great extent when you replace it. All right, so if we inject the virus with GFP in these mice, and then we look in the brain and see how many neurons are getting it. And here in cortex, we see that even if we increase quite a bit the viral load, we kind of hit a plateau here. It's around 35% or so of neurons that we see the expression. And this virus has a synapsin promoter, so most, if not all, neurons should express it. And here's when we put this GFP in our mice, the knockout mice. Again, we get about 30%, 25% of the neurons expressing it. And still, these mice, they get really well once you inject this virus. So when you forget about the virus now, if you just look at the mouse that you delete this gene, right? Because that's a Cre recombinase that deletes the gene in neurons. And when you look the amount of recombination, just looking at the alleles, how many of the alleles recombine. I have to remind you that these are cortex homogenates and hippocampus homogenates. So it's not only neurons, right? So this is a little bit underestimated because you have other cell types that do not delete the gene. But what you get here is about 50%, maybe 60%, will be more in neurons if you had neurons only, right? And the hippocampus is about the same. Now, if we use a different Cre recombinase, another CAM kinase 2 line that's weaker, and we did that, now we get, when we measure in cortex and hippocampus, we get about 20 to 30% deletion of the gene. And when we analyze these mice, no virus now, just the mouse that we delete the gene, you see that the ones that have the weak Cre, they do have less of the nidufas3, you can measure here, but they're pretty much normal. They're not, they don't have the disease that the other mice had. So taken together, these findings show that if you can replace the gene in a certain percentage of neurons, it doesn't have to be every neuron. But just by replacing some of them, you can have impactful clinical outcome, at least in mice. All right, now changing gears to mitochondrial DNA. So we have been focusing on mitochondrial nucleases as a way to improve mitochondrial DNA heteroplasmy. So you all know that you need usually quite a bit of mutant mitochondrial DNA for the cell to be affected. And this can be seen very obviously in a muscle section of a patient with a mitochondrial DNA mutation. You can see that fibers that stay normal for cytochrome oxidase can have very high levels, but not high enough. If you go above a certain threshold, then they get defective. But otherwise, no. So they need high levels of the mutation. That obviously brings the point that if you can reduce the levels of mutant just a little bit, maybe it will be very beneficial. And we've been using this approach that is to express a mitochondrial nuclease that's specific for the mutant and is expressed in the nucleus, but the protein goes to mitochondria and is specific enough to detect only the mutant. And then you have this transient decrease in levels of mitochondrial DNA that whatever is left will compensate. So you end up with more of the wild type. And we've been collaborating with a company called Precision Bioscience that makes this gene editing enzyme called Arcus. It's a monomeric enzyme that detects a 22 base pair sequence of the DNA. And the way Precision does this is that they in silico design the protein, and then they do direct evolution, and they do some cycles of optimization to get a protein that's good for that particular mutation. So we've been working with them. We have been targeting this protein to mitochondria, so we call it mitoArcus. And one of the mutations that we addressed was the 3243, that usually is called MELAS, but actually this mutation is associated with a number of different phenotypes, including exercise intolerance, headaches, etc. So this work was done mostly by Wendy Shoup, that works at Precision. So this is the Arcus specific for the 3243. Here we had a GFP with a T2A sequence, just to show that it localizes to mitochondria. And what Wendy did here was a good experiment, I think. First is to transfect a cell that was 100% mutant for the 3243. And you saw that the levels of mitochondrial DNA here are measured, and you see that they are, they disappear, the mitochondrial DNA, when you use these mitoArcus that are specific for the mutation. This is a control one. And then she did the same experiment with a cell that was all wild-type mitochondrial DNA. And this is good to see what's specific and what's not. You can see here that one of these mitoArcus, the one in red, it also degraded the normal mitochondrial DNA. So it was not as specific. But there were a couple here that were pretty good, did not degrade the wild-type mitochondrial DNA. And here, when she treated the cells with RNA, by electroporating RNA, different levels of RNA, messenger RNA coding for this mitoArcus, you see that at day 1 you have this transient depletion, and this is the respiration. Everybody here respires pretty badly. But at day 3, you see that there is a recovery of mitochondrial DNA levels, and they're mostly the blue type that are the normal mitochondrial DNA. And now, some of those, including these that have a little bit of the blue, respire much better. So that's a nice way to show that this works. And then Wendy tried to do this sort of an in vivo model by putting these MELA cells into nude mice so that you have these tumors, and then injecting different levels of the virus. Now, the tumors do not get well infected with AAV9. The vascularity is kind of low. And you can see here, these are different levels of AAV9 expressed in the mitoArcus, and different tissues here, and these are the titers of the virus. You can see that, the tumor, you see the virus there only at the very high levels of the AAV9, whereas in liver, it picks up the virus much, much more readily. But in any case, when she measured the heteroplasmy, you see that the tumor had some change in the mutant levels at the very high levels of the virus. So there was some loss of the mutant mitochondrial DNA here. It's sort of an in vivo experiment. Now, one advantage of gene editing in general, but particularly for mitochondrial DNA, is that in a nuclear defect, a Mendelian defect, if you want to replace the gene, you have to get expression to be good, not only in the beginning, but forever, because that protein is going to turn over, so you have to continue to produce that protein. But in the case of getting rid of a mutant mitochondrial DNA, you don't have to do that. If you can do it well enough in one shot, and you get rid of the mutant mitochondrial DNA, that's it. That tissue is going to be protected for a long time, if not forever. So, thinking about transient ways to express, we use lipid nanoparticle messenger RNA complexes to express this mitoArcus in cells. And so if you inject lipid nanoparticles, messenger RNA, into the blood of the mouse, what usually happens is that all the expression is in the liver, and this is the liver of the mouse. So either tail vein or retroorbital, these are all IV methods, and that's where the lipid nanoparticle, at least the most common lipid nanoparticles, will end up. Now, if we do this with the mitoArcus, and again, this is against a mutation. I guess I didn't explain this well, but it's a mouse model that has a mutation in the tRNA alanine gene that was developed by Jim Stewart and Niels Larsson a long time ago. So if we inject the mitoArcus in these mice, IV, we see that the heteroplasmy changes a lot in liver, as expected. So it's almost 100% of the mutant is degraded, but not so much in other tissues, maybe a little bit in spleen. So that's usually not what we want when we're addressing a mitochondrial disorder. So the other way to do this is to inject intramuscularly the lipid nanoparticle. And here, this picture shows an example of injecting lipid nanoparticle messenger RNA or lipid nanoparticle DNA into the muscle, in the leg muscle. And you can see expression here. There's always, there's expression in the muscle, that's the TA muscle, but also there's leakage, and some will end up in the liver. And with messenger RNA, it's better than DNA, and seems to last longer, the expression, up to 72 hours here. So we decided to do that. So we injected lipid nanoparticle intramuscular in the TA muscle to deliver this mitoArcus messenger RNA. It's a mitoArcus specific, in this case, for this mouse mutation. 3 hours post-injection, and we inject only in the right leg, not in the left. The left, we inject lipid nanoparticle with GFP. You can see, in this case, we just injected the GFP, not the mitoArcus, just to see expression. And we saw that in the right, you see the GFP there. at 3 hours, and at 24 hours, you still see it in the right, GFP, and you see some in the liver because there's that leakage that I mentioned to you. Okay, here's with the mitoArcus injection. And so we inject in the right leg, not in the left leg, and you see that in the muscle, that's the tibialis anterior muscle of the leg, there was a decrease in the mutant levels, and so close to 50%. Of course, the liver was always more. Just by leaking from the muscle, you get a lot of lipid nanoparticle expression in the liver. But there was quite a bit in the muscle. If you inject that lipid nanoparticle, how far does it go into the muscle? And we want to ask that by taking 2 pieces of the tibialis anterior muscle. And we saw a decrease that was kind of similar between the 2 pieces. So it looks like there's quite a bit spread of the expression once you inject in a single site in the TA muscle. So how long does it last? And we're talking about that in the beginning. So here you can see that this is the TA right leg, that we follow the mice after 1, 2, 12, 18, 42 weeks. And it looks pretty clear that once you get that decrease in heteroplasmy, it's maintained over time. At least, grossly, it's maintained both in the muscle and in the liver here. And of course, these tissues, nothing changes when you inject. This mouse model doesn't have a strong pathology, but has a very clear molecular phenotype that are the levels of the tRNA alanine. That the more mutant you have, the lower the levels of the tRNA alanine. Here is normalized to another mitochondrial tRNA asparagine. And that's because the mutation destabilizes the tRNA here and it gets degraded. So the right leg that was injected with lipid nanoparticle now has higher levels of tRNA alanine. So this tRNA was stabilized because now you have more of the wild type. Another interesting observation we made was in collaboration with Elizabeth Barton at the University of Florida. She's a muscle physiologist. And what she did was to analyze the tibialis anterior muscle in terms of contractility. So if she measured the force, it was really no difference between the mutant and the wild type. But if she tried to look at fatigue, and that means you just stimulate the muscle repeatedly over and over and over, then she saw that the muscle that here's this mouse is the mutant injected with GFP. If you compare the force in the beginning and force after a lot of stimulations, you see that the force goes down. So there's a higher fatigue here. But and this is the wild type, and the treated mice seem to be similar to the wild type. So there was an improvement in the fatigability of this muscle. And that was nice to see because it's hard to have a phenotype in these mice. This is the measurement. This is comparing the wild type left and right leg, and here is comparing the injected with the non-injected leg. All right, it's nice to improve one muscle, but how clinically relevant is that? Well, so we decided to inject several muscles, and in this case here we injected the big muscles of the leg, the quadriceps, the TA, and the gastrocnemius. And after 21 days, we analyzed it, and we saw that there was a decrease in mutant mitochondrial DNA in the big muscles. But interestingly enough, there was also a decrease in the small muscles that are very close to the big ones. So maybe there was a little bit of leakage there that went to these muscles. And this is GFP injected. Of course, there's no change. So you can think that it's crazy to inject different muscles, but people do that in the clinics. There are some muscle problems that several muscles are treated at the same time. All right, in the last 10 minutes I have, I'll talk a little bit about base editors of mitochondrial DNA. So changing gear a little bit. So this technique that was developed in 2020 with the discovery of a toxin that was a cytidine deaminase. So it's a toxin, but if you split it in two, you have two pieces that will only be active if they come together. And they can come together by putting a DNA binding domain that recognize a specific sequence. And if you make all this thing go to mitochondria, you can edit mitochondrial DNA. So we try to adapt this system to the same mouse model that I was mentioning that was developed in Neil's lab by Jim Larson and other colleagues, that causes that point mutation, is this 5024 A here is actually a C to T in the DNA, but it's a G to A in the tRNA. And it disrupts this base pairing, and this tRNA becomes unstable, as I mentioned to you. So when we decide to do this, we were using this base editor that was specific, was the first one described by David Liu's lab, that could change a C that was preceded by a T. So this C would be changed to a T. So we could not correct the mutation with that, but we could correct the opposite base in the tRNA. So the sequence would be different from the wild type, but what we'll do here, by mutating this C to a U in the tRNA, would restore this base pairing here, so hopefully would stabilize the tRNA. And this has been done in evolution many times. Sometimes the primary structure is not so important if you can keep the secondary structure together. So this is what we did. There was Pepe Barrera's lab, a student in Pepe Barrera's work, a student in the lab. And this is the base editor. You have these two pieces here. That's where the business takes place, and this is where the DNA is recognized. And the idea was to change this base here that we call on target. See that the original mutation is very far away, right? But that to make the folding correct. So there are not many other Cs that could be mutated, but there was one here that could be mutated, this C preceded by a T, and we call that, here we call it off target, but so more a bystander mutation because it's in the window that you can edit. So there is the on target and the bystander. So the first thing that Pepe did was to get a cell line that was essentially homoplasmic for the original mutation, because then it would be easier to address if the editing was working or not. So he did some ethidium bromide treatment and got clones, and this clone 3 was essentially homoplasmic for the original mutation. And then he did the base editing by electroporating RNA into these cells. And we end up with two cell lines that we call low on-target editing, about 32%, and high on-target editing is about 78%. Both these clones were essentially homoplasmic for the original mutation, and they had a little bit of the bystander, right? About 16 to 20% of the bystander. The bystander is here. In the beginning, we're not very concerned because there are some species that have this kind of mutation, like humans, for example. Okay, but to make a long story short, then Pepe looked at the levels of the tRNA alanine. I told you that it's very unstable. So these are the levels of the tRNA alanine for the control cell lines. And here is that mutant clone 3, that is 100% or almost 100% mutant. And this is the low editing clone 1, and this is the high editing clone 2. So it seems to be improving the levels of the tRNA alanine here. Then he measured by Western blot the levels of proteins that are quite dependent on mitochondrial DNA integrity, like NIDUFA B8 or COX-1. And you see that the low editing, there was some improvement, and the high editing, there was even more improvement. Here there's an overshoot compared to the controls. And STHA is nuclear coded. There's no difference between all of them. All right, the next thing Pepe did was to make AAV9 and inject this into mice. And so he used different titers of the virus, AAV9, with these base editors. So of course, there are two proteins for the base editors, so there was a mixture of the two. And you see here that at low doses, you get a lot of on-target editing and some bystander here. And as you increase the dose, you come to almost total on-target editing here, but then you get a lot more of the bystander, right? So if you would go for, let's say, a clinical application, probably you should go low so that you get a lot of on-target, not so much of the bystander. All right, so looking at the tRNA alanine levels here, you see that using these low doses was enough to improve the tRNA levels in the heart of these mice. And you can see here that the more on-target editing, the more tRNA alanine levels you had, as expected. And also there were some markers, metabolic markers that improved, like lactic acid, again in the heart, was decreased. Serine, proline, phenylalanine that were a bit increased were also decreased. All right, so now what happens when you increase the dose of the virus? And here we have the low dose, 2x, then 5x, and 10x. And here's the mutation. And this is the sequencing, the next generation sequence of the whole mitochondrial DNA. You can see that with the low dose, you get this on-target in green and the bystander in red. And as you increase the dose, you have almost complete on-target editing here, but then you start to get more of the bystander. And you increase more the dose, then you start to see, you get all these off-targets that you can already see here all over the place. So that's not good, right? And in fact, if you look at the mice that got the high doses, you see that they are sick. You look at their muscles, because AAV9 goes pretty well in transduced muscle. It's very atrophic. If you look at cytochrome oxidase staining in these mice, it's pretty bad, and you see an increase in succinate dehydrogenase, so it looks like a mitochondrial myopathy here. If you look at the levels of tRNA alanine in this high dose that are the ones in red here, they still improve a little bit. But the mouse is sick because the problem now is not the tRNA alanine, but all the other genes that are being affected. And here in the heart, if you look at COX-1 in DuFaB8, you see that's down. Again, not because of tRNA alanine, but because of all the other off-target mutations. All right, so in conclusion, the first point I want to make with the nuclear ones, the replacement of oxidative phosphorylation genes via the AAV-PHPEB, in less than 50% of the neurons can prevent the development of a mitochondrial encephalopathy. So this really we should pursue and try to test in different systems to see if it's always the case, because this has clinical relevance. Now, the mitoArcus is effective in eliminating mutant mitochondrial DNA. So Precision Bioscience was gearing up to do a clinical trial in 3243 myopathies, but the financial environment for biotechs in the US is very difficult, and they had to pause that program. They're focusing on their things that are closer to the clinical trial, that's Duchenne muscular dystrophy, but hopefully they will come back to this when the situation improves a little bit. Then we are also exploring the lipid nanoparticles. They have limitations, but they have some advantages. I mean, one advantage that I can see is that you have a patient, for example, with the 3243 mutation that has a very severe and quality of life impactful myopathy. So this patient might want to treat this myopathy, but if you do this treatment with AAV, you might preclude this patient to receive maybe a better treatment in the future with AAV, because there's an immunological barrier, right? Once you get exposed to AAV, usually you cannot be exposed again. It's not going to work. So you could treat, for example, a patient like this with lipid nanoparticles in muscle, and you still keep the patient naive for a future treatment with AAV. So that's one potential benefit that I see. And finally, for the base editors, they have a lot of potential, particularly for homoplasmic mutations like Leber's the mutations. Now, the off-targets, still remain a problem. People are addressing that, but a lot of the papers I see, they kind of they brush it aside, or they Yeah, they don't consider how hard it would be to control that when you do it in a clinical setting, when it's harder to see how many mitochondrial DNAs are going to be hit by your base editors. So I think this is the main barrier for base editors right now that a lot of people are working on. And I think that's my last slide, so I'm just going to thank the people that did the work. Brittany did the nuclear work, Wendy the melas, Sandra the lipid nanoparticle, and Pepe the base editor. Those are other people in the lab that helped as well, and my collaborators and granting agencies. Thank you.